I'm talking about in situ rolling circle amplification for detecting functional gene inside bacterial cells. And yeah, I start with this slide. Why single cell analysis? Now, I think the bacterial cells like a puzzle, jigsaw puzzle. And I think if you finish them, maybe you never mix them up at first. But in microbiology, we do that. So at first, we mixed many genes from the bacterial cells up, and then you can detect functional gene in the pile. And after that, you try to identify them from this single piece from the pile. But it's very difficult. So what I want to say here is it's very natural that we go with single cell analysis. So the concept of this study. So finally, I want to finish this circle. This circle starts with environmental sample, and you extract DNA or RNA and PCR and sequencing, and you get functional gene database. And based on the database, you can design the probes for detecting functional gene. Then after the detection, you can sort the detected cells by a flow cytometer or laser microdissection. And then you can extract DNA again and PCR sequencing. And now you can get ribosomal RNA gene sequence. And now you can link between bacterial function with identity. And then you can design the probe, and you do a normal fish.